OK, so HIV is one of the scariest pathogens that infects humans today. And I just want everyone to raise their hand if they agree with me that HIV is scary. You'd be scared if you were infected? Yeah, pretty scary, right? And over the last like 20 years or so, a lot of research has gone into how to treat HIV. And we've made massive steps and bounds in treating it. And now it's no longer necessarily a death sentence, but just a chronic illness that does have lots of complications. But that's obviously an improvement over dying. So although HIV is the most infamous retrovirus, it's not the only retrovirus. And it's not even the only retrovirus that infects humans. In fact, every single human being, from cradle to grave, carries inside them, as an inherent part of our DNA, thousands of endogenous retrovirus sequences in various states of decomposition from evolution. So these retroviral sequences have been associated with a lot of diseases, but there hasn't been, until very recently, any really good association between any particular retroviral sequence from an endogenous retrovirus and any particular disease, which means most people just kind of ignored them because they didn't do anything. But now we have evidence that human endogenous retrovirus K is implicated in ALS. And our lab is working on proving that it might be the cause of this disease, amyotrophic lateral sclerosis, which is a progressive, lethal neurodegenerative disease that currently has no cure. So my thesis is focusing on using the techniques and the methods that have been used and developed to fight HIV and applying them to endogenous retrovirus K to create tools that can be used to make drugs that might be able to help treat ERV-K-mediated diseases like amyotrophic lateral sclerosis. I'm focusing on the retroviral enzyme protease because it's been one of the most successful drug targets in treating HIV and also is still active in endogenous retrovirus K, whereas many of its other enzymes are no longer useful because they are needed for the replication process, which endogenous retroviruses don't go through. So I'm hoping that once I have this model complete, this computer model, I can use it to screen drug databases to identify drugs that can then go through in vitro and in vivo validation in order to prove that they are able to inhibit the ERV-K retroviral protease and then be used, hopefully, to treat diseases like ALS.